Hello, everyone, and welcome back to Creation Myths. We are live tonight. So, as always, chat, you know the deal. For a live show, it is your job to tell me if the audio and video look okay. If there are any audio visual problems, just let me know and I will fix them. So, just let me know if everything sounds okay, and we will get into it. Today, we are going to visit our friends at Discovery Institute. And I know I've been picking on Discovery Institute a lot lately, but they deserve it. So we're going to pick on Discovery Institute some more. But to be fair, tonight, we're not just picking on Discovery Institute. We're not even just picking on creationists tonight. Tonight, we are also talking about real biologists who don't understand the basics of evolutionary theory, who misuse evolutionary theory or ignore evolutionary theory of how we are going to talk about tonight. We're going to talk about this piece from Discovery Institute. This is on Evolution News, the Discovery Institute kind of blog thing that they run. And this piece was called Nobelist Thomas Cech on Junk RNA. And that is, I had to look up how to pronounce his name. It's pronounced Czech. There's a decent chance I mispronounce it as we're talking, but it's Czech. So here we are. We're going to talk about Thomas Cech talking about junk RNA and how Discovery Institute presents Thomas Cech talking about junk RNA. We're going to illustrate two things here. First, we're going to illustrate this kind of broad problem of biologists who are not specifically evolutionary biologists, not doing a great job in terms of using, representing, or implementing evolution when drawing their conclusions. And then we're also going to talk about something fun that this piece illustrates, which is creationists taking anything from mainstream biologists that has even just like the slightest whiff of something that they could construe as helping their case, and then promoting it, even when, if you actually read the thing they're promoting, it doesn't actually help the creationist case. And yeah, it is really rich. This is right. This website is called Evolution News, but as Austin points out, it's a super misleading name. It's a creationist propaganda rag. Yeah, that's exactly what it is. It's the Discovery Institute blog, and they usually have multiple pieces per day that they're just a bunch of different authors throw out there. So let me, we're just going to scroll through this. It's not very long. We're going to go through it. We're just going to talk about it, keep it real simple and informal for this one. So as we scroll through here, we're going to jump down here. Those numbers, those are just for me, so I know where I want to kind of look around. So starting here with this line, you can always count on creationists when they find somebody who says, like a mainstream biologist, who says something that they can take as support for something that the creationists want to promote. They'll always really hammer on that person's credentials. So we've got Nobel Prize winning biochemist Thomas Cech. And I just want to start with this, right? Cech, he's a biochemist, and he has a Nobel Prize for his work with RNA, and I believe it's related to RNA and ribozymes, showing that RNA can have catalytic properties, which is a really big deal. That's a really big, important thing. So he's obviously extremely smart. He is way smarter than me, and I do not care. Let me tell you how much I do not care how smart Thomas Cech is. I'm sure he is a brilliant biochemist. I do not care, right? Because when we're talking about scientific claims and hypothesis testing and drawing conclusions, the rule in science is put up or shut up. Okay, you gotta have the goods. Either you have the data on your side or you don't. And like this happens, this kind of thing happens all the time, right? I will say something. I will say genetic entropy is fake, right? And creationists will love to find some random biologist that they can take out of context and say, well, actually, this really famous guy says that the human genome is degraded. Do you disagree with biologists, whoever it would be, right? I think Michael Lynch is the one they love to trot out in that case. Um, yeah, yeah, I do disagree with that person when they say that thing or when they, with the representation of what they that creationists will often provide. Oftentimes that's a misrepresentation, but like, yeah, I disagree. I don't care how famous someone is. I don't care if they're a big name in the field. Bad arguments are bad arguments, right? And like a lot of big names in biology make some really extraordinarily bad arguments that they should know better. They should know make. They'll make arguments about evolution, about sex and gender, or as we'll see here, junk DNA. I don't care how big and important the person is. That's not an argument. So and so has a Nobel Prize. It's not an argument, right? I'm still going to disagree with them. If they're wrong, they're wrong. And as we'll see, like right away, the next thing I'm going to show you all, Thomas Cech is going to do himself no favor in like trying to convince people who know the field specifically that he's talking about and know the history that he is actually well versed when he's opining about these things. Okay, so let's scroll down here to this long quoted section. We're going to go through a lot of this. This is a quote from this piece that Thomas Cech wrote, and then just a little bit from Discovery Institute headnotes at the end. So we're going to go through this. Okay. Now, starting right here, this top paragraph right here, you can see the highlighted section. He's talking about basically, what's in the genome, and I see one person saying that there's audio issues. Anybody else, chat? Let me know. Everything on my end. Oh, okay. We're saying there are audio issues. Okay, stand by, everybody. We're gonna fix this. Hang on a second. Let's go to settings. Let's go to audio. Okay, that's looking fine. That look okay. It all looks good for me. We're gonna keep going. It all looks okay on my end right now. Let's keep going. So he says in this highlighted section right here that if you take away the protein coding regions and the introns, so the introns and the exons, that leaves about 75% of the genome that they consider dark matter, right? Like it's not protein coding, it's not part of a protein coding gene and then removed later. And he says for decades, this 75% was dismissed as junk DNA because whatever function it had, if any, was invisible to us. Like, that's wrong. Like, right away, right off the bat, he gets the history wrong. And that is an enormous red flag. I've talked about this at length. It comes up all the time. Creationists always saying this about how, we just assumed that anything that wasn't protein coding was junk. There has never been a time in the history of biology when we thought all non-protein coding DNA was junk DNA, right? I've talked about this. I've done a video on this. It's linked down in the description. For example, right, we knew that tRNAs were a thing. Those are it's transfer RNA. Those are short little bits of non-coding RNA that are critical to the process of translation of mRNA and using it as a template to synthesize a polypeptide. We've known about transfer RNAs since before the term junk DNA was defined in the scientific literature. So there has never been a time when we thought that all non-protein coding DNA was junk, which is to say did not have a function. Yeah, Andrew Cumming. Also, I believe bacterial promoters have been known about for a while as well. Yes, the lac operon, for example, was elucidated prior to the coining of the term junk DNA. Right? You have the very famous papers breaking down the function of the lac operon, how bacteria like E. coli regulate the metabolism of lactose. Right? We figured out that regulatory system that involves non-coding regions like promoters and operators. We figured that out prior to the junk DNA being defined in the scientific literature. So clearly, we knew that there have been non-coding regions, regions of the genome that don't code for an amino acid sequence that still have a function, right? It has never been the case that we thought all non-protein coding DNA was junk. So what Thomas Cech says here, right off the bat, this, and like at this point, I'm going to be skeptical of anything else he says about junk DNA because he doesn't even understand the history of the field and where the term came from and where it sits relative to the rest of the development of the field of genetics. Okay, so anyone who makes this claim is either ignorant of the history or lying. I think for creationists, they're mostly lying, but for someone like Thomas Cech, he may just not be well versed in the history of the field and that term. So now we're going to talk about this long section here, and we're going to go through a bunch of these highlighted regions, and then we'll talk in some length about this part because there's a lot to talk about in this section right here. Okay, so he says the total number of RNAs in the dark matter has been estimated to be several hundred thousand. These are not things like messenger RNA, but they're non-coding, right? So same categories, ribosomal, transfer, or telomerase RNA and microRNAs, but what they're doing is still, for the most part, a mystery. And this is a key point that Thomas Cech accurately says we found functions for some, we don't know the functions for most, the vast majority. That's true. He says in a few cases, they clearly have a biological function, and he describes a couple of them. But such verified functions are few and far between. The function of most long non-coding RNAs remains unknown. Yes, that is true. We're going to come back to that line in a little bit because it's relevant to something the Discovery Institute author says down below. So Cech continues, as a result, many scientists do not share my enthusiasm for these RNAs. They think that RNA polymerases, the enzymes that synthesizes RNA from DNA, make mistakes and sometimes copies junk DNA into junk RNA. Yes, that is accurate. Now, I take issue with the as a result because it's not just that we haven't found the functions. That's a problem here. But I want to finish this section and then we'll come back to it. But as a result is problematic because it's not an argument from ignorance like creationists like to claim. And Cech is doing real biology no favors by feeding into that here. It's not an argument from ignorance. There's a very specific reason that we say the rest of these sequences probably don't have a function. Okay, let's continue. It says a more scholarly description of such RNAs might explain them away as transcriptional noise. The idea being, again, that RNA polymerase isn't perfect. It sometimes sits down on the wrong piece of DNA and copies it into RNA, and that RNA may have no function. I readily admit that some of the long non-coding RNAs may in fact be noise bereft of function, signifying nothing. Okay, thanks. However, I'll point out there was a time, not too distant past, when telomerase RNA and microRNAs and catalytic RNAs weren't understood. They hadn't been assigned any function. They too could have been dismissed as noise or junk. Okay, so let's talk about the problems with what Cech is saying here. And again, I want to remind everybody that Cech is not a creationist. He's, a well-regarded mainstream biochemist, right? So the situation in terms of the prospects for functionality in the rest of that stuff is actually much worse for like junk DNA deniers than Cech represents here, right? Not only do we have very few long non-coding RNAs with documented functions, as he correctly describes, right? Few and far between. There's a handful in the various classes of long non-coding RNAs where we can document this specific sequence does this specific function. There are a few examples of that as you go through, signs and lines and herbs. You can go through and find examples among every category of specific examples that have particular functions. The problem is not that we haven't documented a function for the rest. The problem here is the remainder can't do any of the functions that we have so far documented. Because the functions that we're talking about, these are things like regulating transcription. That's when you have a DNA sequence and RNA polymerase makes an RNA template that processes transcription. RNAs can regulate that. You can also regulate with RNAs, you can also regulate translation using a messenger RNA as a template to make a peptide sequence through a ribosome, right? You can have another RNA that regulates the rate or even prevents that process by interfering with the messenger RNA or in some way disrupting that process. We can also have long non-coding RNAs that have immune functions, right? They'll help the immune system fight pathogens like viruses. And they can do this in a couple of ways, right? They can, if you have an RNA that's complementary to like the viral RNA, then it can grab onto it and interfere with it. You can also have an RNA that's the same as the virus, and it basically tells the immune system what to target, right? So there's different ways that your long coding RNAs can have an immune related function. All of those functions have in common the fact that they require specific sequences, right? If you don't match the virus correctly, you're not going to have an immune function. If you can't interact with the mRNA in some way, you're not going to regulate translation. If you can't bind to the DNA, you're not going to regulate transcription, right? So all of these different activities that have been directly documented, these, I should say, functions that have been directly documented that Check is talking about here, they require sequence specificity. And the vast majority of long non-coding RNAs don't experience purifying selection. And folks, I love my people. Good job. See, you all know where this is going. Purifying selection. That's exactly right. Purifying selection is the problem here, right? If you have sequences that don't experience purifying selection, you're not maintaining any specific sequence for that mRNA or for that RNA molecule, that long non-coding RNA. That means they don't have a function that requires a specific sequence. And I'll give you a specific example that we can use right here. If we go up a paragraph, right back up to this paragraph right here, we have this long non-coding RNA right here, TUG1, right? It's essential for mice, male mice, to be fertile. Okay, that's a great example because that requires a specific sequence of RNA. And if you have deviation from that sequence, then that particular mouse will be infertile. That male will not be able to have kids. That means that the RNA sequence that's wrong, the one that's deviated too much from the functional sequence, that won't be passed on to offspring. The male can't have kids. That's purifying selection at work, right? You have a sequence that does a job. If it deviates sufficiently that it can't do the job anymore, you don't reproduce, then any sequences that are too far off from the functional sequence won't persist in the population. Ta-da! Purifying selection, and it keeps the sequences the same. Why does this matter for the question of how much junk DNA is there in the human genome? It matters because we've directly documented that only about 8% of the human genome experiences purifying selection. That's a fairly well-known paper. It's linked down in the description to the title of the paper, so you'll see it. It's down there. Um, so you can claim that, like, hey, we found all of these functional RNAs. You know, it's not a lot, but it's some, right? We have all the rest of these that we haven't found a function for. We expect to find a function for. Okay, perfectly fair to make that argument. That is perfectly fair to say. But it's not okay to say that they're going to do the same functions that we've documented because we know that all those other things are not experiencing purifying selection. So if you're going to say that they're functional, you have to propose some other function, some function that doesn't require sequence specificity. What are those functions? Right here we have, right here, Laserfan 17. Are there any sequences known to be functional that don't experience purifying selection? Yes, sort of. There are sequences that don't experience purifying selection on the sequence itself, but where region needs to be a certain length, right? We've got these spacer regions in our genome where you need to have this thing and that thing, right? Like a promoter and an enhancer. These are elements that regulate gene expression in the genome. They need to be separated by a certain number of nucleotides because the way that works is proteins will bind to the enhancer and then the DNA folds over, and then those proteins affect the promoter. And it has to be the right distance. If it's too short or it's too long, that hairpin doesn't work right and you lose the function. So there are sequences in our DNA where you have purifying selection to maintain the sequence in place. You can't delete it, right? You'll have a cost if you delete it. You can't make it longer, you can't make it shorter, but the actual sequence doesn't really matter very much. So that's a thing. Um, but like we can measure that, right? We can identify those sequences, and that's not going to apply to the sequences that we're talking about here because we're talking about sequences that are transcribed to RNA, and then the RNA goes off and does something, right? So that would be a completely different thing, separate from a DNA sequence that doesn't have sequence specificity, right? So when this comes up, I love to ask creationists. I say what I just said about how these functions have to be non-sequence specific, right? They have to operate without sequence constraint. I say, what are the functions? And I've never gotten a good answer to this question. And like I have directly asked creationists, like when I had my debate on junk DNA in whenever that was in 2024, May or whatever it was in 2024, when I debated Dr. Casey Luskin from Discovery Institute on junk DNA, I asked him this question directly, and he gave me a few examples that all require sequence specificity. He gave me examples that don't apply. Right. So I've never gotten a good answer to that question of, okay, what do we propose those sequences to be? Or what do we propose those functions to be that aren't experiencing purifying selection? Now, there's another problem that comes up here that is not specifically mentioned in this quote or in this Discovery Institute article, but I want to just kind of mention it anyway because you hear it all the time when you hear this thing. And it's the argument that you, again, often hear it from biochemists that it's the idea that processes like transcription are energetically costly. It's true, it does cost a lot of energy to do transcription. So if a cell is doing it, it must have a good reason for doing that. Like there must be a function associated with that energetically intense activity. It wouldn't be wasting all of the energy on spurious transcription. But here's the thing, evolution predicts spurious transcription as long as the cost of that transcription is below the selection threshold, right? Because you've got natural selection shaping genomes. You also have non-selective processes like genetic drift. And if the cost of the presence of a DNA sequence or the transcription of that sequence is sufficiently low that natural selection can't see the difference between an individual who does it and an individual who doesn't, if that difference is too small, then selection will not be operating on that transcriptional activity and won't clear it out. It won't select against it, right? You won't have selection in favor of the organisms that don't do it. And it has been empirically shown that this is the case for most transcription. I've linked that paper down in the description as well. So if you want, you can go down and click on it. It's a really interesting paper. It's called The Bioenergetic Cost of a Gene. And basically, the idea here is that spurious transcription is expected to be extremely common because, relatively speaking, the cost of that is quite low. On the other hand, spurious translation, right, junk proteins, we might call them, are expected to be very rare because that is much more energetically intensive for the cell. So the cost of making polypeptides that don't have a function is typically above the selection threshold, so you can efficiently select for mechanisms that prevent that. So what we expect to see is lots of transcription going on that doesn't actually do a job, very little translation that isn't associated with a specific function. Those are the predictions, and that's exactly what we see in actual cells. So Transcription itself, and this last point I want to make before we move on to the next part here. Transcription itself cannot be taken as evidence of function without some other corroborating thing, right? Without some other corroborating feature like a high level of transcription, purifying selection, something, right? Creationists love to claim, you hear this all the time, that transcription can be taken as prima facie evidence of function, and that is wrong. And it's wrong for very specific reasons that have been empirically documented, right? Now, if you're making that claim that transcription indicates function most of the time, they're either unfamiliar with the theory they're criticizing or they're lying about it. And I think when creationists say it, they're mostly lying about it. I think when biochemists say it, they're probably just not super familiar with the evolutionary implications of what's going on under the hood here. So I know that was a long section we went through. So let's move on to the next part, which is a little bit shorter here. This little bit right here, part four. I just want to point out this line. The problem is that it is a presupposition that could be holding back scientific process. And you hear this from Discovery Institute all the time, that the evolutionary paradigm gave us junk DNA, right? And we would have tossed away the idea of junk DNA long ago if it wasn't for those stupid evolutionists, taking over the field. And if people had the freedom to look for functions where the evolutionists said there weren't any, then we would have documented this stuff already, right? Now, I mean, this is wrong, right? Like, first of all, the human genome was only sequenced fairly recently, within the last 25 years, right? So it's not like you've got decades and decades and decades of history of people saying, don't do this, don't do this, don't do this. Unlike, stomping down on people who try to investigate the genome. We've only been able to investigate the genome in a specific way for the last, at most, 25-ish years, right? But like this whole idea that evolution is holding the field back is just wrong. You know how we know that? Because it's mainstream scientists who almost without exception, almost to a person, accept mainstream evolutionary theory who are making all these discoveries in terms of the functions of, endogenous retroviruses that have been co-opted and long non-coding RNAs. It's not creationists making these discoveries. It's like real biologists, right? So if this was a thing, if evolution was holding back the field, if it was preventing people from doing this work, then the only people making these discoveries would be the creationists who said, no, we expect to find function there because the genome was created to be highly functional. But here's the thing, creationists don't do real research, right? When was the last time you read about a creationist getting in the lab and getting their hands dirty and actually doing an experiment to show like how something works? That there's actually function in part of the genome where the mainstream biology said there wasn't function. That never happens, right? All these papers, like in code, for example, that creationists love to tout, those are all mainstream biologists who all, to a person basically accept like the basics of evolutionary theory, right? The fact that we found the new functions at all refutes the creationist claim that evolution is preventing us from finding these new functions. Like, it's not more complicated than that, right? It's silly. All right, so let's keep going, right? Now, this is the Discovery Institute author. This is Daniel Witt writing this here. He says, as far as I can tell, Check assumes RNA will have function simply from experience, not from any underlying model or paradigm. In contrast, other scientists don't share his assumption because they, like Check, are working in a paradigm that predicts junk, evolution, right? And unlike Check, they form their expectations based on that paradigm, not on the emerging pattern of evidence. So I want to bring this back um, to what I said above, right? The few functions that have been found are not relevant to the remaining RNA, which is the vast majority of it, right? It's not about we find function here, therefore we expect to find it here, because here and there are two different sets of sequences that have different characteristics, different types, and different levels of activity. So as I said before, and I'm not going to belabor the point, but as I said before, if it's functional, it's going to have to be different functions. You can't just extrapolate what we found and say the rest of the genome does similar things, right? You got to find something that doesn't require sequence specificity. And with this second piece right here, this part's really important, this in contrast. This part's really important because, uh, like, the people who are realistic about junk DNA, who acknowledge that most of the genome is junk DNA, we're the realistic ones here, right? We're not saying that because of like an underlying evolutionary paradigm demands it. We're saying that because our predictions are accurate in terms of what we've talked about, in terms of things like spurious transcription versus spurious translation, in terms of like the level of purifying selection in the genome. It's just wrong. You don't have to be more complicated than that. It's just wrong to say that the idea that junk DNA is real is based on a paradigm as opposed to the emerging pattern of evidence, right? The evidence is that we do find some functions in these sequences, and then the rest, we can identify those functions. It's not like we haven't looked yet, folks. We're not making, like I said earlier, we're not making an argument from ignorance here. We're making an argument based on, okay, we found these functions. We've looked at the rest. Those sequences are different. They can't do those functions. And in fact, we would expect them to still have this biochemical activity that we observe in the absence of a selected function. Now, creationists are going to keep on saying it anyway, right? They're going to keep saying, the trend marches on, right? We're just going to keep making this progress. But they're going to keep saying that because what creationists are doing is a confidence game rather than like genuine scientific inquiry, right? For their target audience, repetition is enough to keep people in the fold, right? The point is to just repeat it, and just through repetition, it's going to get through to their very specific audience. So I want to scroll down just to this last little bit here, right? It's increasingly clear that the junk DNA narrative is old. The older generation of scientists may not be ready to confront the implications of that, but the next generation will. First of all, I'd like to think that I'm still young and I'm part of the next generation. So we will see. We will see. Um, But this is an example of creationists citing sources that don't help their case. It's increasingly clear that the junk DNA narrative is over. When we hear that documented functions for long non-coding RNA is few and far between, that does not scream to me the junk DNA paradigm is over. That screams to me there may be slightly less junk DNA than we thought 30 years ago, right? Maybe it's 5% instead of 90%, right? That's what that says to me. That's a far cry from the junk DNA paradigm is over. And I will say again, this is not just creationist fault. This is creationists running with the ball that they're being handed by like biochemists. So for example, actually here, we'll look, I can highlight this comment from Nafi Khan. I'm in a Discord server where everyone, including multiple PhD biologists, are extremely hostile to junk DNA and insist that it's debunked. That's completely wrong, and we'll follow here. Thank you for your comments, by the way. My brother said the same thing from PopSci articles. Yeah, exactly. It's exactly it. It's been really bad reporting, and a lot of it stems from just a few papers that Encode put out. Like a few papers, like one big paper in 2012 and some subsequent like press releases. And that has led to this whole thing. And there's not a robust foundation for that conclusion. Discovery Institute, or sorry, not Discovery Institute, and Code subsequently walked back some of their conclusions in a 2014 paper that I always like to highlight, where they laid out the conditions and said, if a sequence meets these conditions, then we think it's probably that's a good indication that it's not doing a selected function. And then in the data of that same paper, like most of the activity they talk about meets the conditions that they laid out for probably not functional, right? So there's a communication issue here among extreme biologists, some of it driven, by money, some of it driven by what I think is good faith misunderstandings of the evidence or being told something and kind of credulously accepting it without checking it yourself. But I want to go back up here because I want to highlight this line from up here, such verified functions are few and far between. Again, I want to contrast that with how Discovery Institute is portraying this. The junk DNA narrative is over. Now, if you read that line, the junk DNA narrative is over, you probably think that we've documented a function for something like 80 or 90% of the genome instead of like somewhere in the neighborhood of 10, maybe 12% if you're being generous, right? Creationists will actually admit this if they're kind of pressed on it, right? So when I debated Dr. Casey Luskin on this question of junk DNA in 2024, he admitted that it's the standard that he said is it's less than half of the genome has a documented function. It's way less than half. But I was like, is it half? And he's like, no, it's less than half. So okay, great, it's less than half. Your organization, right, the Center for Science and Culture, right, from Discovery Institute, they're saying, oh, junk DNA is dead. Most of the genome has a function. They're not saying we found a little bit and we expect more. They're saying it's dead, blanket statement. It's completely at odds with the reality of what we've actually documented. Completely at odds, right? So I think what's going to happen here, we talk about the next generation. I think what's going to happen here is like people are going to get wise to what's going on with this junk DNA story that you can't just say over and over and over again, we're going to find more functions, we're going to find more functions, we're going to find more functions. Eventually, given the fact that we have the sequences for the complete genome, you're going to have to start finding specific functions for this stuff, or people are going to stop accepting that there are actually functions there. And I think that's what's going to start to happen. I think in spite of years of PR spin from like Encode and then like a lot of creationists that have picked up the Encode stuff, the evidence is what it is, right? We're looking at these sequences biochemically and in terms of their activity in an evolutionary context. And the actual conclusion is very clear. Most of that RNA, despite being active in some biochemical sense, isn't doing anything affirmative for the cell, right? There's not a selective function there that's helping the cell survive, right? And one thing that would help biologists, mainstream biologists, real biologists, as opposed to creationist hacks who are just going to lie about whatever. I have no hope that creationists are going to ever admit anything vaguely correct related to this. But I think a basic grasp of evolutionary theory would go a long way towards mainstream biologists who are not evolutionary biologists, cell biologists, biochemists. Uh, it would go a long way towards helping them make sense of the data and the distinction between a biochemical activity and a selected function that's maintained through natural selection, right? When you dig into it, it's clear that most of what we're seeing is activity without a function. And I think Thomas Cech is a good example of somebody who kind of falls into that trap. And then we have here this article from Discovery Institute, despite taking Cech's accurate representation, right, that it's not that many we found function for, but then running with that and extrapolating wildly to the idea that, the junk DNA paradigm is dead, right, which is ridiculous. All right, so that is where we are going to end this, folks. Basically, with my plea to all biologists to learn some basic evolutionary biology, it will inform your work. And if you think that the field of evolutionary biology is not relevant to the study of like biochemistry or cell biology, here is a great example of how that is wrong, right? How a basic understanding of evolution will prevent you from being led astray in these much more micro focused fields, these molecular fields, right? So that is where we're going to leave it for tonight, everybody. Thank you all for watching. I appreciate you all being here. I appreciate fantastic comments. I really love my audience, all of you here watching live that you guys, you follow along with this stuff and you know exactly the beats where I'm going with this. So thank you all for being here and for your contributions while we were going through this recent blog post from Discovery Institute. So as always, before you all go, please remember to do the algorithm friendly things. Hit the like button, leave a comment, If you're here watching, you just stumbled into the stream about junk DNA, please subscribe and enjoy your stay. Share this, see what Discovery Institute thinks. Send it to Thomas Cech, see what he thinks about this random person saying he's wrong despite his Nobel Prize. Thank you, everybody. Stay healthy. Have a great night. I will see you next time.